Okay, so let me see. So I guess you can see the whole thing, right?
Yeah.
Mhm. All right, so first page, committee members and Prano, advisor. I mean, this is strange. You put your name and you put and suddenly advisor and your email. So this is confusing, right? You don't necessarily put the advisor name here in this
Okay.
case. So, I mean, like this. You see my point? You don't need.
Yeah.
Maybe you can
Yeah.
make it bold.
Okay. Okay. Here, yeah.
Then it's distinguishing, right? Right.
Okay.
Okay. And how can I clear stuff? Annotate it. Close. Oops. Okay. Good. All right. Somehow. Okay, annotate. Yeah. Clean. Is there any clean? Clear. Clear or dry. Okay. So next slide. Okay, this one. Content. So in general, like intro, literature review, method, results, and this summary. I mean, this is generally okay, but I don't like this way of writing content. The reason is this is not specific. What it means is if you put this way, like every 100 presenters will have the same. So it's no fun and nothing special, and people would not learn anything from here, right? So my suggestion is you better to have a specific way. If it's introduction, then introduction to what? You need to always consider the theme. So here, deep behavioral phenotyping of diabetic neuropathy mouse model using open field test. This is a little too long, but this is a title, right? Title originally proposed, right?
Yeah.
Okay. And neurophotonics lab, this one, okay. This is a little too dim. Okay. So I have one thing from here too. So I guess I have to tell you about this. I can see that you're trying to put as much information possible. This one is, I guess you're trying to put diabetic neuropathy that may affect only like foot and hand, like distal limbs. So that's probably diabetic neuropathy. And you want to put MOSIC. This is, looks like the MOSIC company, of not company, but the labs logo. And this is part of the MOSIC.
Yeah.
Okay. So, but and this is mouse. You're showing that this is from the mouse. In general, like putting this many objects is not the best idea. I mean, this is to me like, wow, there are many things. And out of this, I don't see any figure you actually have created. You're borrowing others.
This one?
Is this you've made?
No. I just take from another.
Yeah, because the basic rule in academia is when you are taking someone else's, you should have a reference or citation. Because in academia, the credit's very, very important. And you are using others' figure without any credit. And probably this is too typical, so probably okay. So that's why I don't think it's a good idea to put like, unless you have a real key diagram you want to put as a background would be fine. But this way, it contains information, but in general, if I see, it's not beautiful. So, and this is not even your own creation, not your own. I can have information from the mouse here, but if I read, people read diabetic neuropathy, people with proper knowledge can guess what's going on. So my suggestion, I mean, these are cool, but that may come from somewhere introductory part when you are kind of describing your approach. This might come there. This may come somewhere because you already said mouse model. I don't think it's necessary here. This one too. When you are describing diabetic neuropathy, somewhere introduction, you may use this one. Then people catch, okay, in nervous system, this affects this. But not necessarily in the, I mean, this first title is the most important. And we are already seeing someone else's drawing, which is not great. We are already just giving copies of others' work. Okay, so that's my opinion. May take a look at that. Okay, so, and clear all the drawings. And let's move on. Yeah, this one. So whenever possible, that's why I put all the defense materials you see in my office, in front of my office. There's a cabinet. I put all the stuff. You may go take a look. There are good things, good examples there. Because this, let me see, back here. You would probably put introduction to blah, blah, blah. So you have to consider what's the core concepts, core message. What are you going to introduce here? Okay. So think about that. Literature review on what topic? Okay. I mean, the order is fine. Method sounds a little bit, approaches. In general, like you would put, I think you have a nice hypothesis and approaches. And you have results and discussion and summary, that's fine. But this is just non-specific, and in general, I don't like this. Because you can copy and paste anywhere this, and there's no information here for us as a reviewer. Okay. So that make it specific. Also, if it's methods, then you want to put something here, some more information. Okay, sorry. So if it's method, what kind of method? I think you are using like kind of MOSI, some specific method, which is very specific. So you want to describe method, or I guess it's more like approaches or introduction and maybe problem definition. And what kind of problem you are. So if it's methods, you want to put like 1, 2, or 1, 2, 3, so that it can be very, very specific. So that people actually look at it, oh, this slide has information, and expect what's coming. But right now, this is just too general, and we have no idea what's coming. Do you agree?
Yeah, yes, Professor. I will be more specific.
Okay. Let's move on. Next. Yeah. So now introduction. Like I think to me it looks like introduction. Sorry, the font is a little not good because I'm putting into a keynote. I'm opening this one, so I think it's fine. So like in general, like you don't want to put just introduction. I mean, there are contents here. Introduction to what? I think it looks like problem of diabetes, right?
Yeah, diabetes.
You want to put like kind of more specific. But I think this one, IDF, if I look at it, I cannot find anywhere what this is. I mean, this is actually what frustrates readers. When they look at it, they have to wait until you say. So international diabetic,
Yeah.
like something, federation or what, association. So always put acronyms, you put it somewhere, okay? But the purpose of this looks like a little bit, I think you are talking about like this one, diabetes is caused by hyperglycemia. There's a lot of people suffer from diabetic neuropathy, and you put normal and diabetes nerve. Fine, blood glucose level, international. I mean, this is not a point. 1 out of 8 adults will be suffering from diabetes, and the source is IDF, I guess, right?
Yeah. Yeah.
I would, yeah, IDF. Then you would put it like you have to be specific on 1 year, like 2021, or I mean, the source has to be clear. When you are talking a number, don't put this way. Then we cannot see whether you are rigorously having the proper sources are here. Heart, kidney, nerve. So when I read it, like, okay, I mean, this will be affected, right?
Yeah.
And out of this, the nerve is what matters for the neuropathy.
Yeah.
So it's a good drawing, but the drawing is like when people look at it, like blood glucose level, then probably, I mean, it's probably elevated blood glucose level. This is what? This is like maybe incidence. It's a prediction of incidence, right? And this is affected organ. Largest global epidemic of 21st century. I mean that's probably relevant to this, but is this like, is this true of what you say? I guess maybe some of these references may have, because people may disagree, like, hey, diabetes is not the most global epidemic. Someone can say, hey, obesity would be global pandemic. Someone can say, how can you refute that? Is this something you say or something it has a evidence?
This one is evidence in the at the end of the slide. I put in CDC.
Okay.
CDC diabetes.
Then you would put like double quote, okay?
Oh,
and then
oh.
CDC and some report 2000 blah, and you would put that CDC, yeah. Reference. You have always reference there. Okay. I can see some, but in general, you just put introduction, so what? Rather, you want to put your title out of this. What's your message here? Like, because I don't think you want to just explain what's diabetes is important. I mean, everyone knows it. That's nothing new. You want to draw attention for diabetic neuropathy is coming and it's important to resolve. That's your take-home message, right? Then you put introduction, maybe blah, blah. The problem, the emerging issue of diabetic neuropathy, something like that, specific. Then people, oh, okay, right. Otherwise, it looks like, hey, everyone knows diabetes is important. Why are you talking about diabetes to spend this one whole slide? Sounds good? So you want to make this introduction more specific and make this take-home message clear with the references. Do you agree?
Yeah. Yes, Prof.
Okay, let's move on. Again, introduction to what? And now you're talking about diabetic neuropathy more.
Neuropathy,
yeah. Pain, motor function. Morbidity means quality of life, understand. Yeah, this would be a good place for diabetic neuropathies, like incidence with numbers. Understanding mechanism of DN, diabetic neuropathy. You want to put diabetic neuropathy in novel therapeutic approach, risk and protective factors. So this is for what? What's necessary for resolving this problem of diabetic neuropathy?
Yeah. Understanding the pathophysiological mechanism of diabetic neuropathy,
Okay.
they have importance for the three things.
Okay.
First, for novel and identify risk and protective and biomarker. Like I thought this one is I would explain when I
Okay.
talk to when I present. I will explain this thing how it is interconnected.
Okay, I see. I, I don't think you need the figure 1, figure 2, figure 3, because it's not a paper. You're not
Mm,
necessarily making numbers of figure 4, 5, 6. Or is there
yeah.
any reason for that?
Uh, of,
Because you have a
for what?
list of figures? No, all these figure.
Yeah, this one because I want to explain to the audiences what
Yeah.
behavioral syllables means first.
Yeah, but it's not your figure and your paper figure. It's just figure here, right? I don't think you need to put figure 1, 2, 3, 4 here.
Mm,
It's not necessary because figure should be confined to one slide. That's all.
okay.
You don't need these figures number, okay? Unless you put a list of figures. But these are like, I think these figures are also coming from internet or somewhere other sources.
Yeah. I put the reference figure at the end of the slide.
Okay, okay, let's, which one? Let me see. Do you
All
have all
figures.
the, do you
Yeah.
have all the sources of the figure?
Yeah.
That's why you
Yeah.
put it
Yeah.
there? Where?
Yeah.
Where?
At the last.
Oh,
Last.
I see.
Yeah. This.
At least you have figure numbers. Okay, that's fine. Okay. In general, like when you put this one, you put that figure source over here. That's more
Mm,
proper. But anyway, you have it backwards, so I think that's fine. Diabetic peripheral neuropathy. This is not peripheral neuropathy. This looks like a treatment of
mm.
diabetic peripheral neuropathy. And you put somewhere from, and then drug that can delay onset of type 1 diabetes. Okay, people may ask about this drug because given that you put it here. Okay.
Actually, this one I will remove it. It doesn't have sense in this one. I would
Okay.
like to explain the drug, what
Yeah.
they did in a recent report, this one,
Yeah.
but maybe
I
I will
mean,
remove it.
yeah. Given that you put this, meaning that you know about what you put, you are supposed to be able to explain. But I'm not sure whether we are talking about the drug, specific drug for type 1 diabetes. It's probably
Yeah,
not an
yeah,
important topic
yeah.
here.
Yep.
Okay. But the take-home message for here, diabetic neuropathy, motor function. So you want to put motor because that's one you want to show. Intractable pain, you have a pain assay and obvious sensory loss there. Okay. But this one, diabetic neuropathy, is understanding pathophysiology. This is not a pathophysiology. This is like we need a novel therapeutic approach because it's a problem severe. And
Yeah,
there are
yeah.
risk and protective factors. I mean, this is more like clinical aspects, right? And we need
Mm,
the biomarkers and diagnostic tools and therapeutic. I mean, if from this, I would put this is the first, this is second, this is necessary, this is the
mm,
third. So it's weird that this one comes at the front. I think this one
okay.
should go to the last. when you're making a story out of this diabetic neuropathy problem. Okay.
Yeah.
Let's move on. Literature review. So you have a number of reviews pages. The reason why I asked you is like, you know, everyone in research, you are supposed to have a very exhaustive and thorough reviews of the literature of what's up to date and what are you actually adding on that. So, but here literature review, 1 slide, 2 slide, 3, 4, 5, 6. There's just 6 slides. And this is not very typical ways of people putting their literature review. You can have just 1 slide and comparing what are out there, and you have a kind of summarizing what has been done for this problem. Somehow you haven't defined your problem yet though, okay? So that's a little weird. So when you have an introduction, I can see, hey, diabetes is important, and it ensues neuropathy problem, okay? And then second, so you are focusing on diabetic neuropathy issue, not for human though, but there are pain, sensory loss, motor function. Then you want to define your problem from not clinical, because we are not doing clinical. Preclinical study of diabetes is limited by proper motor function assays. And maybe also pain is also limited. So you want to define problem by use of mouse model of diabetic neuropathy. And there has been limitation for motor function impairment assay. That's your definition of the problem. And for these preclinical approaches, you want to bring out literature survey. And you want to point out the advancement and the limitation of the previous study or emerging new technique, which is MOSIC, right? So that you are adopting that to applying this technique into the specific problem of diabetic neuropathy. That's your story. And okay, so I think this is not a literature review, but more like a problem definition or current status or approaches. I mean, I cannot see any literature here, so it's not literature review. Okay.
Approaches maybe.
Yeah, I mean, yeah.
Approach.
I mean, it's a, so these are like approaches, right?
Approaches, yeah.
Preclinical.
Behavioral approach. Yeah.
Preclinical. Preclinical, yeah. Tests, assay, preclinical methods to assess, right? Preclinical, or it's like this nerve control. This can be even clinical as well, though, but behavior is more like preclinical. Clinical methods to assess the severity of diabetic neuropathy. And you can say behavioral nerve, but we don't have conduction test, right? People will ask about you, why you didn't do it. Then how would you answer? Like we have searched the animal nerve conduction test. There are maybe tools, or you haven't done it, but this can become into your future approaches to make your if area is more complete, people may ask, you need to have all that into consideration. Okay, to assess, methods to assess. Yeah, that's like the title, not literature review. So so you consider behavior very important. That's why you put it top. Histopathology. Don't put this way because this is not good. Histopathological is an adjective, it's not a noun. Histopathology. Okay? Don't put this. This is not good. Okay? Nerve conduction, biochemical assay. So you put behavioral test. Miles or red. Okay, one flay filaments, open field. Maybe you can find something like kind of mouse, and there are mouse foot, like those diagram. Instead of this is, this is tool set. Because you have an animal here, better to have an animal. You see what I mean?
Yeah.
And you can easily find that figure instead of this. Okay. So you're talking behavioral tests, composes of one frame. Are these the only two behavioral tests for the assessment of diabetic neuropathy? Are there any other?
There is another method.
Hot plate and cold plate. So this is for pain, right?
Yeah.
This is not necessarily for DN though. This is more like pain behavior tests. This is for pain, right? And
yeah.
These are for pain.
Yeah.
And this is not specific for pain, but general locomotion.
Locomotion, yeah.
So like behavioral test. You're actually here focusing on like that actually coming from behavior, right?
Yeah, yeah.
So you're focusing, hey, there are many methods.
Yeah,
Are you going to do nerve conduction? Are you going to do any histology? Are you going to do any biochemical? Probably not in your current. So you have to make sure to limit the scope. Okay?
Yeah, yeah.
So from engineering perspective, you focus on this and you have to be able to justify. People can say, hey, these are more important. Why are you doing this? Well,
yeah.
Because this is a study that has a scope, we're looking for more engineering approaches. So we focus on that, right? But this is good to explain and you have to know what's their importance is too, okay? So here you have Bonfray and open field is, so I think you open field motor function coding is good. And hot plate, cold plate. So that's something you have a plan or?
Actually, this one, I would like to explain in this thing. There is hot plate, cold plate, and von Frey test for pain, but the gold standard is von Frey test.
Okay, yes.
That's why we have choose this one.
Okay, I see. So like but people may ask you about like, what about you put it there, why you are not doing this?
Yeah.
Okay. But this is the first step and the most important.
The first gold standard, yeah.
So this is mechanical and this is thermal. And so you don't necessarily say we're not going to do it, but yeah, because the mechanism of mechanical and this could be different. So when there needs a more specific, so for example, if you look at diabetic neuropathy, DN behavioral test, did you find out people have done von Frey? Yes, right? Yeah. Did you find a hot plate cold plate test? Did you find?
Yes, Professor. Some papers do hot plate test as a supplementary to the von Frey test.
Okay, right. So if you're going to do a very serious pain experiment. This is supposed to be done. Okay. However, given the limitation of resources and time, you choose the important one first.
Yeah.
So you can specify maybe future. If you want to do, so the current focus is more like engineering aspect rather than the biology of the pain. So if you find something very interesting and want to go further, then you could, so this could be a future study, right? And you can even combine this one with 3D camera as well. You know, they may jump and stuff, different behavior. That can be interesting, right? So you can actually explain that. You don't necessarily have done it, everything, but that's a future study when your finding is interesting and you want to look at even thermal behavior from this diabetic neuropathy matter. Sounds good?
Yep.
Okay. And literature review. So this is page 6. So again, so not sure whether this is a literature or not. 6, 7, 8, 9, 10. Look at that, Rahano. The fonts are different.
Different.
Is it true? And it's going ups and downs. This is actually very much immature. You want to put it, copy and paste, so the location should be in the same whenever you're preparing. Okay. Well, this is impact of diabetic neuropathy. You're talking about now, you're getting also, I mean, you're spending a lot of material for diabetic neuropathy, like impact, sensory,
This
cognitive,
cognitive, yeah.
So you focus on cognitive here?
Yeah, cognitive.
This one's your focus?
Yeah.
Okay, is this your focus?
Yeah, and cognitive and more about locomotion and exploratory activities here in the above.
So you're talking about human or animal?
Animal. The locomotion and exploratory activities in the upper.
Yes,
yeah.
right.
Yeah.
But this one doesn't look like an animal, but this one shows which it looks more like a etiology of diabetic neuropathy.
Yeah.
That can lead to locomotion effect, right?
Yeah. Feet
But,
and the leg, yeah.
okay, but where's the reference?
I put figure 4, reference figure 4 at
put
the
it
end.
there.
Okay.
Don't put this repeat. Okay, this is enough. Put the reference. Whenever you have, put the reference on site. Okay. And so what are the take-home message here? You have sensory loss, cognitive loss, motor function. So you're actually spending another slides for the problems
Impact,
of
yeah.
I don't like the impact of site. So this is like changes, impact is more like a positive many times.
Positive, yeah.
Okay, so find out proper. So just putting this sensory, like let's say sensory, like a functional loss, cognitive, yes,
Yeah.
cognitive dysfunction
Risks.
also,
Yeah.
motor and coordination, or you can put the problems of diabetic neuropathy or something like that. That's a sensory loss, cognitive maybe. Is
Memory. memory loss.
I mean, cognition and memory is one part of cognition, right?
Yeah. Novel object recognition, YMISS, used for in the preclinical.
Yeah, so maybe cognitive dysfunction or something.
Yeah.
Okay. And motor coordination, maybe what?
Impairment.
Impairment, something. Yeah. Always try to make it clear, not just sensory. So what?
Yeah.
Okay.
Actually, this one, Professor, when I present, it will be better to explain or just put in the name here.
I'm sure you will explain, but
Yeah.
the slide has to be self-explanatory, unless it's too many words. I don't think it's too many words at this slides. So whenever you put OFT, like when there's always a space, open field test.
Yes.
The end is you are repeating it, so I think it's fine.
Fine, yeah.
And don't put the capital letter unless necessary. Okay, like this one's fine because this is kind of original name. But this one,
Name,
no.
yeah.
This one you should use a small capital, okay? Lower capital. But this is not a literature review. You have to put proper title on that, okay? I mean, it looks to me, you are delving into the impairment from diabetic neuropathy, something like that.
Yeah.
So always put the proper title. I mean, this is important exercise. Sounds good?
Yep.
Maybe I'll just also take a picture for you. Okay?
Yeah.
I'll clear that. But we are recording it now, so. Now literature review. So 7, 8, 9, 10. So you are putting like 4 slides only for explaining one paper, which is a little bit rare though. Also, I ask you to prepare literature review because putting 1 slide means you're gonna spend at least 1 or 2 minutes. And that important or not is a question. Okay. So this one, this is the first one. So you have a specific one. So recent paper, that's good, metabolic. And this kind of new two drug combination, experimental diabetes. Okay, that's experimental diabetes. That's good, inhibition of oxidation. So this is about more like mechanism paper and ERG. So that's good. You know, the readers cannot read any of this letter. You know what? Nobody can see this. So whether this is important or not, it depends. So the aim was effect of this drugs. I don't think your focus is about drugs, but you want to point out some essays, motor function, they reverse velocity over distance traveled. Okay, I guess this is kind of the point you want to point out. So control this drug combination, 3 drug combination diet. So here the most important velocity, okay?
Yeah.
And distance traveled or moved. So my suggestion is literature review. I mean, it's not a review, relevant literature 1. And you want to point out experimental diabetic neuropathy. And what made this, how to make this experimental neuropathy model? Is it the same as what we did?
Yeah, yeah, totally.
Plus nicotinamide?
they have used I don't I'm not sure about Nikonima NA, but they have used SZ
Okay, I guess. Is it mouse or rat?
mouse.
Okay, so we are using mouse. So the reason you put this literature is probably like you want to put what kind of mouse model we are using. What were the key literature which we learned from them? The original mouse model for diabetic neuropathy. Here, I mean, these are important, but nobody can see it. So what you want to point out is the limitation of, in this field, the assays are only like two of this in behavioral assays. Okay, that's what you want to point out, right? So
Yeah.
that's too few. Because this drug is not too important for us, right?
Yeah.
This
Yeah.
point. But this is, but given that this is for at the first literature you want to put, I would expect you put the diabetic neuropathy model and the protocol for that is maybe if it's common, then this is a mouse model of DN. And we are adapting that. That's one. And behavioral assay, they have limited one. But unfortunately, this is not possible to read. So my suggestion is, I mean, why don't you move this one down? There's enough space here and remove that, okay?
Okay.
And this is fine. And then, I mean, this one, we can read that this, you can reduce this by this, or make this figure this big.
Okay.
And then, I mean, make another one for this Y so that people can see this velocity, and this currently is too small, nobody can read it. Sounds good?
Yep.
Okay, yes. Another literature, I don't know why this changes, but you want to put literature number 2. And why did you choose this one? So this is another antioxidant, another drug treatment, streptozotocin. And what are we learning from that? So you want to put crossing, so another behavior.
Another behavior.
And the rearing behavior.
Yeah.
No significance. So, okay. So again, this is very hard to see. I mean, the comment is the same, but if we say just crossing segments, we don't know what does it mean. And do you mean by you put the field into like maybe 4 segments?
Yeah, in segments, and then what are the number of they are moving through segments? So there was no any significant.
Like A, B, C, D, and then when they are moving.
Exactly.
I mean, then you should put this exemplary figure as if somewhere. I mean, that's the most important. People would not understand what are you talking about. Okay? So this is another new aspect of behavior. Okay? And then number of rearing. So like people don't know what rearing is. You should have defined rearing. Okay? Definition. And figure of rearing. So you're talking about the mouse like standing up?
Yeah, standing up.
With 2 legs?
2 legs, and then
not necessarily holding on the wall?
This one is the mice. Actually, this one I put in the last slide, but the mouse is just in upward. Yeah.
Yeah, then put the figure or photo, like clear one. So you want to put,
Yeah.
You want to put cross segment crossing,
Yeah.
segment crossing, and rearing first. I mean, you have to explain that first. This data is nothing, okay? You want to put the
Okay.
rearing segment and rearing behavior because we are focused on those two. Put that one here, okay?
Okay.
This one.
Okay.
And then move this data down over this space. I mean, there are too many words. Nobody want to read the words these days, okay? But then put a number of rearing, crossing, then people realize, oh, this paper had behavior only crossing and rearing, and it's not even significant. So that's a limitation of this one, right?
Yeah.
Two things to learn. One paper uses only a couple of behavioral because it's so painful. That's your point to make here. Okay, and even no significant. Maybe it's if we have you have to put the limitation of the study for each. Even the
Yeah.
one, like only two
Yeah.
were two behavior essays were performed. Okay, that's a limitation. Here is also another, only these two. I don't know if they have another, but so limited number of behaviors, and then maybe why it's not significant.
Yeah.
Of course, maybe this drug didn't work. But
Actually, in this slide, the previous this one,
yeah,
and in the next also the literature review slide,
yeah,
I would like to explain. They have used in the same parameters, even rearing and number of cross segments.
yeah.
In the other study, they also showed significant difference.
Okay, yeah,
And also total distance and velocity.
yeah.
In the other one also, like they showed also significant. Like this indicates that like maybe it is inconsistent result with these behaviors.
Okay, yeah,
Maybe the over inside of
yeah. Those are the limitations. Do you want to put the others work? I mean, the point is not the drug study, but the point is there is a limited behavior study, and those are inconsistent.
Consistent, yeah.
And probably that's due to the limitation of the techniques of behavioral study. Okay, but I mean, crossing and rearing, people have to understand at this slides. Okay, so that's the point you want to make here. So literature review 3, right? I think every literature you want to put kind of small, short title of take-home message. I mean, that gives you have, people can see what you are trying to say here. Experimental neurology. You don't need to be this big for that. Open field test, here is cross segment. So now cross segments. What is this?
Yeah, number of fields, yeah.
Is that also cross segments?
Yeah, yeah.
So this is a rat.
Rat model.
So this is different. I mean, this is the same, using the same one, right?
Yeah, this was in the rat. They also show significant difference.
Okay, that previous one was mouse, and I mean, this is basically the same as the previous.
Yeah, the same as the previous.
Right. The only difference is a rat, right?
Yeah.
Then you don't need to put this much. No need to spend this. Literature review, maybe you put this open field, but number of fields, this is not very clear. But number of cross segments, right? Here.
Yeah.
So this is a control. What is DM? N and W and N. When people look at it, people are trying to understand what's going on here. So this is diabetic neuropathy, right?
Yeah, neuropathy.
Modulation by nicapone, this N, antioxidant property. What is this W?
This one, it is without like the diabetic neuropathy without treatment.
Okay. And is treatment?
Yeah, it improved. After that, it a little bit improved.
I mean, this doesn't look like improved though.
Yeah, but they said it is significant.
No, that's not meaningful. I mean, you can look at that. This is not that different. I mean, this is different from here and here. Then
Yeah.
this one, it looks like not significant. Okay.
Yeah,
so,
but we only want to this one and this one. We don't want to about the treatment. Yeah.
Right. So, Prahanu, I don't think this
Yeah.
slide is necessary. You're
Okay.
wasting time. Okay. I don't think it's necessary.
Okay.
You're not adding anything here. It's the same. The only difference is red. Okay.
Okay.
You don't want to. I mean, you can still put this one into move it to supplementary. People ask, hey, you did only just mouse. People can be curious, hey, does this work for rat? Then you put the title, literature review, rats. Okay, this is a rat. Okay. So you don't necessarily throw it out what you made. Each slide, once you make it properly, you can be useful to moving into the supplementary slide. Okay. But currently, this is nothing relevant. Already you've done. So, okay, literature review. Now this is properly like third. So again, neuroprotective and blah, blah, combratin A, combratin B in mouse. So what are the differences? So now it's again rearing.
Yeah.
Crossing.
And here they showed significant difference here. There is in the normal control and diabetic.
Yeah, normal control and various treatments.
Yeah.
Okay, I don't think it's also necessary. You can go move it to supplementary.
Okay.
I mean, the point of literature is unless you are making like a real table, okay? This is like you're supposed to spend a minute or the very precious one and just showing that, hey, this one works. It's the same as before. You want to point out what are the previous problems or limitations. There's only rearing and crossing. So that's only the few behavior. And these are limited and sometimes it's inconsistent. That's all. So it's the same, right? It's not necessary here because your point is not antidepressant works bad, good or bad.
Yeah. Yeah.
Sounds good?
Yep.
So instead, you should review Mosick paper. I don't know it's here or not. Where's Mosick paper?
I put in the motion sequence paper in the discussion part with our results. I discuss.
This one? No.
No, in the discussion. Yeah. Next. Yeah.
This one?
Yeah, this one.
Why are you putting this?
This one, for example, when we compared the traditional methods with the motion sequence methods for identifying pain-induced behaviors in diabetic neuropathy mice, they also consistent with the previous results for drug classification. These motion sequence methods are like they classify drug based on distinct behaviors. So versus traditional methods.
I thought your work is mainly based on mosaic sequence. Or,
Yeah, motion.
right,
Yeah,
then that's the most important literature because that's a breakthrough technology.
yeah, the breakthrough technology.
This is 3D behavior, another one.
This one I would like to connect how the 3D behaviors can connect to the neuronal level like that.
Mhm. Okay. I think your literature review should actually explain at least one full nice slide with a mosaic so that people understand what you are actually based on. Because the limitation is only segment and crossing and rearing number. That's like very limited study, total distance traveled. That's from the literature. So you are introducing a new technology which is emerging very recently.
Yeah.
And this has the capability, and you want to adapt this to apply for this diabetic neuropathy.
Yeah.
I mean, this could, people could consider, hey, that's not that novel, but I mean, adopting this is a big deal. So this is not a good way to actually introduce paper, like just copy and paste. Not a very good way, unless you also want to like maybe highlight some places to focus. Okay. But let's go back. I mean, you have literature review here, like 3. Okay. Limitation. So yeah. So that literature review, probably you have 2 or 3 left, right?
Yeah.
You don't need to like overlaying one. You don't need to waste time for that. So then current limitations on what? Okay. On assessing behavior in diabetic neuropathy models. Okay. Just make sure. So you're, but what is this? Spousing basically kept design post challenge. Oh my gosh, it's hey Brown, you're not gonna supposed to write down full sentence here. You want to make everything bullet, okay? 2 or 3 bullet points. And these references go downwards, okay? Because it's not important for people to actually look at it, okay? So what you want to put is current limitations. Actually, this is not current limitations. This is a emerging or current methods, right? Popular methods.
Yeah, this one figure 9 Ito Vision video tracking software. On the previous slides have used this software.
Okay.
They have a predefined behaviors.
Okay, I see. So that's a limitation.
Yeah, complex behaviors can be challenging here. And then,
yeah. And then you want to say, okay, Ethel Vision. Yeah, this is video tracking, and this is only 2D, right?
Yeah, 2D. And then complex behaviors can be challenging here.
Yeah, complex behavior such as what?
For example, mice naturalistically they express to the world with the 3D pose dynamics to their surroundings in spontaneous behavioral activity.
Okay, then those are like more important to put it there. Mice is surrounding 3D pose dynamics. So you want to put one line with 3D pose dynamics and you put examples, okay? E.g., you know what? Okay, 3D dynamics such as rearing. Rearing is already done, right?
Yeah.
What examples do you?
Actually, sometimes when we see the video, like even rearing in with a 2D thing, they say rearing, but it is not like when in detail. There's very complex. There is
Of course. Okay. So yeah. Okay. Even rearing is not, but I think you want to have a very clear examples for 3D. Okay. You should start with like the previous one, existing methods. Okay. 2D parameters. Okay. Position, velocity, contour. That's one line. Okay. And the second line, you want, in fact, there exists intricate 3D pose dynamics, not possible to capture. That's the second. Okay. And then this is a famous one, the software, the predefined behavior, complex build can be challenged. I think you want to say like some example, 3D, blah, 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 something here. And another, but you have to have Dimlet code is very important. Where's the reference for that?
Yeah, I put here.
Yeah, but I think you want to put Nature Neuroscience reference too.
Yeah, okay.
Is it like 2015? So
Yeah,
check
I
that
think so.
out. Okay. But this is a breakthrough technology. It's very good for detail. But you want to put current limitation. So dimlet card, is it not for 3D, right?
Yeah, it's not
It's
for
image based.
image based, yeah.
But are there any
It is pixel information. For example, in deep lab cut, firstly you have to mark what you want to track. Because if you want to track tail like that, you have to mark, and it is subjective, the experimental mark. The second thing is it is also gives in a pixel information, not the not giving you the video with the pixel, the results. So it is inconsistent.
Sounds a little bit not very convincing though. I mean, this is very great technology, right? But so
yeah.
you want to put the good thing first, okay? This is source first, like this is deep learning based, right?
Yeah,
So
supervised deep
yeah.
learning.
And you want to, yeah, supervised learning. Is motion seek unsupervised?
Motion sequence unsupervised.
Right. So I think the point is there. Deep lab cut is, I think this one should have be about 1/3 of this size. Not that much.
Yeah.
Only about that. Okay, that's enough. But dimlet cut is a breakthrough technology.
Yeah.
I mean, this is really cool. But this one is supervised learning. And did you find anything like diabetic neuropathy model by use of dimlet cut?
I didn't find using deep lab cut for diabetic neuropathy model, but for another disease, autism, like.
Yeah, but remember that the animal locomotion while working on a fear, dimlet cut can do a very good job for like kind of this motion of gait. Remember that?
Yeah.
I mean, that's very cool. That can be applicable. Right? Do you remember that nice movie, Animal? While this wheel rotates, the gate. Well, we are not trying to tell dimlet cut is bad. Dimlet cut is also a very important technique. But it's a supervised learning, so it may put bias, right? Compared to unsupervised. There's some limitation. And I'm not sure whether you would emphasize on that. You should be the author of dimlet cut when you're reading this. Okay. So it's supervised, so that requires an effort. And also, this is not necessarily 3D. This is like based on 2D video.
2D, yeah. 2D
so,
video, yeah.
So that's a limitation based on 2D video information. Right? That's obviously,
Yeah.
I mean, 3D would provide more rich information. So, I mean, this is great technique, and I'm sure we can also use it too, right?
Yeah.
So we are not excluding it, but we are more focusing on 3D. That's the key point. That's why you need another slide for actually the power of MOSHI too. But given that, we have this. Let's see. Okay, I think we have already spent more than an hour. Okay. And I didn't see your hypothesis in the first slide. You should have hypothesis in the contents. See? Hypothesis. Okay. Your hypothesis that don't put the full.
Too much?
No, I mean, you don't want to put a slide, don't put the full sentence unless you are quoting others. People do not like to read the whole sentence when hypothesis on supervised learning with a 3-depth counter capture. Circular open filter will enable subtle dynamic locomotion. Hypothesis is a testable or not. So subtle dynamic locomotion explorer.
Yeah, we have to test because of it's the frequency change, so we can test based on their frequency.
Yeah, hypothesis and aim, right? So given that limitation and literature survey from the previous, I would put your hypothesis dang and (3D) depth camera with machine learning. Okay. You want to put a nice title for that. And so this hypothesis already, so don't repeat. Unsupervised machine learning with a 3D camera. Unsupervised machine learning with a 3D depth camera can, okay, its hypothesis can capture 3D pose dynamics of DN mice in a circle open field. I mean, that's one line, and you want to put to enable discernment of this behavior changes, okay? Too long, so make it two. Sounds good. And now the new concept of behavioral syllable. Okay, you want to define it not here, but here, okay? Yeah. Make it italic because syllable is a very important concept now, okay? Brief, 3D motif of behavior. Oh my gosh. That's repeated in a sequence. It's not very clear to me though, 3D motif of behavior that is repeated in a sequence. This is not very clear to me. I mean, you want to define the behavior syllable in a more clear way, okay? I mean, it's okay, brief 3D motif. But then, like, what is motif of behavior? Okay, maybe repeated in a sequence. Sequence of what? because it is sequence of behaviors.
Yeah.
It's already behavior.
Yeah, it's behavior.
I mean, it's
In
not
sequence,
clear.
in time interval, in a temporal.
Yeah, please make this one more clear. Okay, it's not very clear.
Okay.
And you want to put an example. Always make the quality of the image good. This is not acceptable. This is bad quality, okay? So no reversing, grooming, folding up, grooming self, rhythmic twitching of head, nose is slightly elevated. So you are now talking about syllable, partial reversion, pause, okay, full reversion, pause, and turn. Okay, find out high resolution image. And this is an interesting one, but nobody can read it. So you want to put this one here, and you want to make this large, like this big, okay? Otherwise, we cannot read this, okay?
Okay.
And you have to highlight. Why don't you highlight some important points here? Like if this is syllable, you want to highlight.
Okay.
To me, it's a little unclear though. No reversion, full reversion, partial reversion. I'm not sure syllables is a reversion or grooming or pause and turn.
Yeah.
I mean, you want to define syllables, then you want to
Syllables
have,
in the example.
yeah, you want to have very clear syllable name, like kind of putting here, okay?
Okay.
This the syllable. Then people start to understand what you're gonna say. Oh, this is a new concept. And this one requires maybe 3D, especially like this rearing.
Yeah, this is a mosaic example.
Right. So I think you put this in mosaic, right?
Yeah, nature.
So this is a problem that people can consider as a problem because you are using the code of mosaic paper.
Yeah.
And you're not making it very clear. Then people will consider, hey, have you developed these new ways? Probably that's not true, but you are giving that impression. That's also not accurate. That can become a problem. People are asking you, what's your contribution? Did you make this by yourself? Probably not. You're utilizing what's out there, but we are adapting it to apply it. This is an issue for credit. And then, many people can point that out. And you may give a wrong impression that you have done a great work. Of course, this is not easy and great work, but you're not giving enough credit to the original inventor.
Yeah.
That's why my suggestion is instead of removing those two literature, you want to put the mosaic and the key concept of mosaic. And I'm sure this is actually already a kind of concept, but this syllable is not necessarily need to be in your hypothesis. Your hypothesis is like this, and maybe some why this 3D depth camera would be good. Why unsupervised? Because you talk dim record is supervised, so it's painful.
Yeah.
And it can be depending on the inaccuracies of the people. But this is automatic. So you can emphasize on that aspect for this would be hypothesis. At the same time, this is an approach that you are using mosaic. And I don't see even mosaic here. Right? And people cannot understand if you say mosaic. You have to explain one place that this is motion sequence, blah, blah, blah. Initially. Okay?
Yeah.
So let's move on. So now you're specifying, okay? So this is approaches. And this is a mosaic, isn't it?
Yeah.
Oh, no, but this is not actually mosaic. You're like getting out somewhere. I don't.
This one is, I would like to explain the unsupervised, like that. The input data, and then the model, and then the output. This is the camera.
This is the camera.
Yeah,
So
this one, no.
So this is not aim, this is an approach. And
The
what are
aim
these
is this
three?
one. Yeah,
So specific
already
aim
said.
is evaluate 3D small scale behavioral motifs in DN mice. Okay, it's a specific aim. 1. 2, identify pain-induced behavioral feature in DN and their transition. So that's 2.
Yeah.
Did you have a comparison with 2D parameters? Have you done?
Yep. Yeah,
Okay.
yeah, I have
so,
done.
So that's a specific aim, but this is approach and specific aim. For you, approach and specific aims. This is aim 1, aim 2, aim 3. This is how you want to put it. Okay. So approach is you are using unsupervised machine learning plus 3D depth camera. Right?
Yeah.
Why don't you put like kind of a little more specific, like Kinect, like 1000, something like that.
Okay.
Because this camera looks the same as anything else. Nobody knows whether it's 3D or not. And this one, I'm not sure whether what you are trying to say is
this one just unsupervised machine learning algorithm to see the media.
But to me, it doesn't look like unsupervised. I mean, it could be supervised. Why is it unsupervised?
No, for example, unsupervised, when you give input data and the model process the data and then they give you output.
Right.
You don't have to be labeling or something, like just input your data and
but
then
this
the model do. And
model may be already supervised model.
Yeah, the model 1 is based on the data. They teach based on the input data. Yeah, but don't require human intervention.
Okay. Yeah. But this look like human brain, though.
Yeah, like that. Yeah, the model looks like human. They teach by itself.
Yeah. To me, it's unclear whether this one can automatically tell me as an unsupervised. Maybe supervised, you have like kind of maybe some more labels here, but it's unclear. Why don't you find maybe a better one showing just unsupervised machine learning?
Okay.
Please replace. I don't think this is good enough. So you said evaluation 3D small scale behavior motifs in mice. Evaluation of behavioral syllable, right?
Yeah.
And that syllable is defined as 3D small scale behavioral motif, isn't it?
Yep.
I mean, this is too many words. So evaluation of behavioral syllable.
Yeah, we can see that.
Indian mouse. And then you put like this. And then you put here, what is behavioral syllable is defined as, like in your case, in our case, 3D. I'm not sure whether small scale behavior is proper words. Please check that it's a proper words. But behavioral motifs.
Actually, this one I took from the definitions I took from them and also other papers.
Okay. But you have to be able to explain why it's small scale. And you need to be able to say, well, then what's the motive? Okay.
Yeah.
Because motive is also need to be defined as well. Always make sure define things which is coming out first, because it's specific here. Okay. And
Okay.
so now it's identify pain is behavioral feature in DN and their transition. You're talking about now two things,
Yeah.
feature and transition.
Yeah.
Okay. But behavioral feature is also a syllable too?
Yeah, syllable.
So now it's a syllable too. Okay.
Yeah, syllable.
And in this case,
yeah.
the context here, this syllable in context is behavioral feature. Is that different from left one?
It is the same, but we have to identify,
Yeah.
like with the diabetes neuropathy induced. They have changed after that. First, we identify the syllables.
Yes.
After that, we have also identified the changes. They
Yeah,
will put
changes.
some
You're
syllables.
talking about your change. Do you mean transition or like specific syllables, specific for DN mouse?
Okay, here, but first, aim. First, we have to identify
Yeah.
3D small scale behavioral motifs, syllables in the in our DN mice.
Diabetic mice, yes.
Diabetic,
That's
yeah.
identifying which ones are
Identifying.
maybe important.
Yeah, identifying.
But
And
second is identifying.
Yeah, second is identify the pain induced. Yeah, like
yeah. So first one is more like application of MOSIC to this our model.
Yeah, exactly.
Right, application of MOSIC in our DN mouse model.
Yeah, for example, here we get 22 behavioral syllables in the first aim.
Yeah, this is 22, right?
22. In the second aim, only reducer to if we identify 1-4.
How many? 14.
14, yeah. And then the second one means transition, means from one behavior to another behavior also. So we have to identify that.
So that means a 14 square transition.
Yes, transition.
A to B, B to A.
Yeah. That also change, whether it is change in the DN mice, we also did that.
The third is 2D.
Yeah, 2D or usually we say traditional parameters. Yeah. Position,
compare
velocity.
3D with 2D parameter.
Yeah, but without motion sequence.
Yeah, 3D usually possesses 2D, right? Yeah,
yes. That's why we in 3D camera we have also recorded with
automatically
2D parameters.
Right. Yeah. So like it's really not comparison. It's like finding out what are the additional information of 3D compared to 2D, traditional 2D.
And then after that, we compared with the logistic regression.
Okay. And then what did you find?
We find that in the result, motion sequence identify using motion sequence outperform these traditional methods in our study in DN model. Okay, yeah.
Okay, so I think aim one is more like it's like, you know we learned about MOSIC, we apply MOSIC, apply in our mouse, diabetic mouse model, evaluation, application of behavior, evaluation. Well, it's fine. You are not putting too much of MOSIC emphasis, while throughout this is MOSIC, right? Evaluation of behavioral syllables in DN mouse model. Mouse model, okay? Okay, and aim two is now you're applying to find out pain induced. So this is, in fact, it's application, whether it works or not.
Yeah.
So you apply this. This is from MOSIC, right?
Yeah.
You apply to see if it works. This is testing whether it works or not. And after it works, you pain-induced now behavioral features. You want to find out correlation with the von Frey.
Actually, this one we can say for correlation, but in the diabetic mice and control mice.
Yeah.
And then see their difference in usage of these syllables.
Right, but yeah, but you starting with the out of 22, you compare correlation of these syllables, whether there's a positive or negative correlation.
No. Only control and
DN.
Control and DN.
Right.
Usage.
Yes, and you found 14.
Yeah.
It's different. So this is outstanding, like important motives for diabetic neuropathy.
Yeah.
So the aim one, you identify, apply this to DN versus control.
Yeah.
To identify important syllables. That is like 14. Like we reduce it. 14 syllables are probably meaningful in specifically DN mouse model.
Yeah.
And then your aim is to analyze it further, not only that syllable, but also you found transition might be important.
Yeah.
Right?
Yep.
So you identify, actually this is kind of coming here, identify pain-induced syllables.
Yeah.
Behavior feature, I think you want to put like kind of here, like parenthesis and syllable.
Syllable, yeah.
And this is evaluation of, I mean, this is too much. This is too much. I think this one, you put behavioral syllables so that people can see syllables over and over. Because this is an important concept. And you explain what are the syllables defined. And their transition, like nobody understand when I read it, their transition. Okay, syllable transition, what? Pain-induced syllables. Identify pain-induced syllables. You don't want to put the end initially, the back pain-induced syllables and their transitions in diabetic neuropathy model, in DN model. Okay? Make sense?
Yep.
The third is you want to compare 3D, like this approach. And this approach's name is MOSIC, isn't it?
Yeah, this approach is MOSIC.
But does the MOSIC also include a transition analysis as well?
In their analysis, they exclude the transitions, but they compared with traditional methods.
Transitions.
No, they didn't do transition. They excluded.
Really? They didn't do transition analysis?
Yeah, transition analysis they didn't do.
So you are telling the transition analysis is come from other paper?
The transition analysis comes from other paper.
Yeah. So I think that's important point. So MOSIC and so that's like syllable and so identify in pain, syllable and syllable transition, two concepts is important.
Yeah.
Syllable is coming from MOSIC and syllable transition. Did you put that transition reference?
Yeah, I put their references, the transition one.
And did they use for diabetic neuropathy or something else?
They have used for another animal model.
What? Is it for diabetes or pain? What?
I think they have used for the transit of how the naturalistic behavior with the DLS, like at the neuronal activity level, at the neuronal activity level, they like DLS neuron, like they impacted this neuron and they see their transition is affected.
But that's neuron. We are talking about behavior.
Yeah. They also see these syllables, their transition is affected by that neuron.
For what model? Are there any disease involved?
Not like they have no as naturalistic behavior. They didn't do disease model.
I mean, that's why it's that paper should be in the literature as one slide. So because you are proposing concept of, if it's a syllable, then you are just using MOSIC and applying to the end. That's all. But with that, another literature, which has not been used for disease or diabetic neuropathy, you are the first to utilize that to apply for DN mouse model. And so that's important literature to show that it's not just about syllable, it's about syllable transition. That might be also important in behavioral aspects of this pain related, or it's even if it's not pain necessary, but yeah.
Actually, in motion sequence paper, they first did transition, but in their paper they said they excluded their transitions in the paper because they didn't find important. So in their drug classification, they excluded it.
Okay, fine. But then using the transition, okay. Anywhere is fine, whatever. But the original paper for that transition is important.
Okay.
So that people can recognize, like it's not about just syllable, it's about syllable transition.
Transition, yeah.
Especially for this disease involved. Why don't you try to put that as a reference too? I mean, that's important. One is MOSIC, that is about syllables. The other is transition of this.
Transitions, yeah.
That's the their transition and syllable transition.
Syllable transition. Okay.
Sounds good. And the third is now the comparison of approaches. Logistic regression. I mean, that regression is not the purpose. Your purpose is like comparing why we need a 3D depth camera and this approach maybe outperform or than the conventional compared 3D.
Yeah, traditional is best. Yeah, that
or traditional. 3D versus 2D. It's not. We're not comparing parameters. 2D approaches. Okay, and the specific parameter can be something. You can actually put this main, and you can have a bullet points like here, like this. Okay, because there's only a space. And please don't make this mistake. You don't want to make this. You want to make it professional. Okay. Okay. And now I think I'm going to just point out the quick things. Brahano. So, I mean, this is good to have. Glucose meter for blood. So look at von Frey testing. Lab scale. Why are you putting lab scale? So it's a body weight. So open field test. Mouse. Behavioral tests. Okay. For mouse. For diabetic neuropathy. mouse. So this is important, baseline. I don't know where the 0 point, somehow there gonna be some 0 point. Why it starts with week 2? Where's week 1?
Week 1, we didn't do anything. We just we just inject the strategy to the.
I mean, that's the point. You inject STG. Where's that point?
Yeah.
Why I don't see that?
Yeah, I just only the behavioral measurements here. I put.
Okay, so
I didn't include.
Brown, this is very confusing. Okay, so baseline you should do baseline, but is this different time point, different days?
No, in the baseline as the same. Different time means yeah, as the same date.
First of all, I have to point out, I think I'm repeating to you, like, is this very careless?
Yeah.
Okay, then never do that. Okay.
Yeah. Okay. Actually, this one I will change it because I already did
You already changed
the
it?
alignment. Yeah, I changed this one.
Okay. And the point. So usually at this timeline, study behavior. I mean, timeline. This is experimental protocol, right? Timeline.
Yeah, protocol. Yeah.
Experimental design. They're not only just time.
Yeah.
I mean, this is kind of design what you have, but like when you put like how many N, how many animal?
Yeah.
How many animal?
12,
Is it only
12 animal.
one or what?
No, 12. 1, 2.
What?
1, 2. 12.
Please say 12. You talk
12, yeah.
total 12. How many
Yeah.
DN mouse?
8.
How many control?
4.
DN mice. Usually, okay, you put 8. Okay. So how many weeks old when you start?
This one, the open field, you mean open field test?
For these tests.
3 weeks.
3 weeks old? No, I'm talking about the age of the
the
animal.
mice age. Okay, I think. Let me see the. I don't maybe
Okay.
8 weeks, 4 up to 6 weeks.
No, please make sure. Okay. So for example, like 8 to 12, it's okay if it's a range high, but narrow is better. Okay.
Yeah.
Or
Okay.
male or female?
Male.
Okay. Male. That's also a problem because if you're not doing female. Okay, so that's your mouse mother. Okay.
Yep.
And then control n equal 4, and that's the same. You have to make sure this part. Okay, so that's experimental design. Please make sure to double check that. Okay.
Okay.
The height this is not that important. Okay.
Yes.
This is, you should have a separate slides for people will ask you, how did you use the Kinect with the open field? What are the stage? Because you didn't, here it's not very clear, right? And this has to be shown the bottom. Okay.
Yeah.
Instead of this. Okay. Flip it 90 degrees. And computer is not that important though. Like, why is it this emphasized? I mean, this is the most important in terms of size. So this is baseline. We are doing one fray test. Okay, one fray. Maybe you don't need T, one fray. Glucometer, this is a blood glucose level, right?
Yeah.
I think that's what you put. Lab scale is body weight.
Yeah.
I mean, not that important, but you should have all the data. Why did you do only twice of body weight? So once a week?
Yeah, once as the baseline one time,
Yeah.
and also at the start of 2 week only one
Okay.
time.
So you have only two points.
Yeah, 2 points.
Do you have data plot for
Yeah,
this?
yeah,
Okay.
I already plot in the result.
In here?
Yeah, here.
Okay. I mean, body weight is just basic. You don't necessarily put it there, in fact. Okay. But if you have a data or nice data of the week, so baseline weight, right?
And week
And
2.
2 week,
2 weeks. Yeah.
week 2, body weight. So you have a control,
Yeah.
okay? And you have diabetics. If it goes down, then it may become a problem, but it's better to be like, people want to see this, okay? But for you, when you're doing this, better to at least measure the body weight every week, because body weight changes and it's the easiest one to measure. Okay, so easy one to gather data. So, I mean, this is a little, like ideally I would put baseline. So we need to put once week 1, 2, or before you sacrifice, or if you are sacrificing it, something like that. So it's a future, okay? But people would expect baseline. So you don't need this way, one fray, blood glucose, right? Level and body weight.
I see.
You don't need this, okay?
Okay.
So baseline doesn't need to be that long, but the importance is day 0. When did you inject? I mean, you should need STZ,
week 1.
ascasic, and nicotinamide if there is. Okay, so that's week 1. Week in fact 0. Baseline, you put it there. You need to put NTZ injection. Like,
Yeah.
how did you inject? IP?
Yeah, IP.
And you need to have the dose for that. DN mouse, NTZ, how much? Let's say, what is it, 10 milligram per kilogram body weight, something like that. Hold injection, IP, something like that. You should describe that here. I guess this one was done by you or Asik.
Ashik.
So later, make sure you credit Asik that what he did. Because otherwise, you will be bombarded by question. Did you do it? You have to make sure, give that credit. Okay? So week 0, baseline is before, week 0 antigen injection. And then you wait for 2 weeks, right?
Yeah.
1 week, you haven't done anything.
Yeah. 1 week we only injected.
Yeah, week 2, or did you specify the date? Week 2 is just too long.
Yeah, we after like 2 weeks from the baseline at
So
the end of 2 weeks.
So what date? Did you have a specific date?
Yeah, we have a specific date. Yeah.
Like, is it from 10 to 14 days or like 14 days plus minus?
I think that we have did this one in the week 2 from Monday up to, I think, up to Tuesday, like that. But we have a specific date. I will add.
Please write down. So days, it's not this is so short. You are not talking about week. You have to say about day.
Yeah.
Okay. So you have to say day 0. Day 0 is this STZ injection.
Yeah.
That's day 0. Before you measure this baseline, that could be like, you can say day -1. Or you don't actually, this is baseline, so it doesn't matter too much. So your experiment protocol should be something like -1 or just before. It's usually okay. But day 0, and this is day let's say you want to say 2 weeks, like 14 plus minus, but you have multiple animals, right? Plus minus, let's say 2 or 3, or you want to say day 13 to 15. And then you have measured these one altogether. Again, Bonfrey, I mean, it has to have an order. Blood glucose level is first. Like, I don't know which one's your order, but to find out whether it's diabetic or not, and then Bonfrey,
Okay.
and maybe body weight.
Okay.
That's the week 2. What did you do week 3?
Week 3 or open field test, because we have allowed rest for agitation.
Okay.
So in that week,
do you
Monday,
have a day or something?
I will do in 3 days, from Monday up to Wednesday.
Okay. So it's maybe a good idea to say 20 to 23, something like that. Or this is fine. You can put week but so when you say week 0, week 1, week 2, 3, you put specify date.
Okay.
Dates. This way you can be more precise and people can see how rigorously it has been done, because week is just too wide, right? But this one, you didn't do other things. You did the open field test.
Yeah, open field test, yeah.
Okay, then you want to move that open field test somewhere here, okay?
Yeah.
And computer, reduce the computer small. I mean, this is open field. I mean, that's a good thing. You have this and Kinect and to watch, okay?
Yeah.
And body weight. Then you can distribute it. This is a little bit not like the most important thing is blood von Frey. If you want to von Frey, you want to, so like that test. Open field test. You have all details of this, right?
Yeah, I have the next slide. We have a detail for that.
Great. Okay, so experiment. This is the most places people will bombard you question, and if you don't know the specific, you are out. Okay, so this is the most important to make sure. Okay, let's see. Oops. Let me clear. So this method. I hate this. Please put some proper title.
Okay.
Methods just too broad. Okay, so you're using this specific one. You know what? This one has a reference too.
No, this one is original.
No, I got this one from Singapore and I gave it to you.
Yeah.
We created. So there were paper for this. Kinect.
I see.
Let me. So, okay, this is kind of detailed. Good. So, Rahanu, please remove all these ones.
Okay.
Ask someone to do this for you. Okay. Or just retake by putting like white paper outside. Yeah. It's okay for tomorrow, but please do this. This is not good. This is unnecessary, right?
Yeah.
Okay. Depth camera, that mouse. I mean, this one's good to see inside, outside. There's a specific information for this. Let me see.
This thing. Yeah. I want to explain here the height and width and something.
Yeah, I understand that. That's fine. But you know, this is not good. You want to move this one in the middle. Why are you moving here? Why are you making this an empty compared to this?
Because in order to for my case, in order to align this one,
Yeah.
I like put the first figure to the upper one.
No, that's not
left.
that important.
Okay.
You want to make it design the arrangement. Right now you are doing this. Okay. Well, that's not good. You want to put middle, or you want to spread it out. So make the line, left line and right line the same. That's an order.
Yeah.
Okay.
This one, but the second figure is very too long.
I know this is
Maybe
too long.
yeah, the resolution when we compacted, the resolution was very low, I'm afraid.
I know what you mean. But there's a, do you have the original schematic of this? There's a
This one,
there's a like AutoCAD file for that. And the
this one I
reference.
did. This one I did in PPT.
So you can put the CAD file or something so you can fill that, because this design is important. And you didn't put the Kinect information. I mean, this is the key. Why you're not putting the more information for that?
Yeah, I didn't put it.
Yeah, I mean, that's the most important. So you want to put the Kinect. I mean, this is just hardware. Kinect is important. Okay.
Yeah.
And some of the key feature, maybe a couple of that. Frame rate and the ranges and blah, blah. I mean, that's the key here, right? And the bottom one, please make sure the line is aligned. This is not aligned. Okay. Acquire data. And you said that you spend like 15 hours of data. That's a lot of data.
Yeah.
But that can be emphasis. You say in one session, like what's the time for that?
30 minutes. 30 minutes for each.
Yeah, 30 minutes for each. And that's one session per session.
Yeah, per session.
So out of this experiment design, how many sessions did you actually collect?
Actually, if we classify session, for all mice, for example, for 12 mice, one time recording, let us say session 1. So I did 3 sessions.
Okay, yeah, but there are like various animal numbers.
Yeah, various animal, yeah, exactly.
Right, and groups. So
yeah,
you can compute how many sections, let's say 100 sessions, for example. And one session, what's the data size? Like, let's say 100 megabytes per session. So total, maybe you have 1 terabyte. And those informations are very important for conducting and understanding what's going on or how difficult it is, how much data set you're actually generating. So that space, you can actually use that, acquire arena detection, extraction. It's not very clear, compute principle. In fact, this figure is kind of not understandable though, but that's fine.
Yeah.
But you want to make the size at least comparable and remove this strange border unless you are doing the same way, okay? Extracted amount. What I mean, this one, this is unnecessary. Yeah. But that line should be aligned properly, okay? That line. What do you mean by extracted mouse? This is like extracted or adjusted, right?
Yeah, extracted from the data. Yeah. We got this extracted mice without even without noise here. But this is our data.
But is this the first and then you extract it?
This one is the video extraction.
But this one's after you have a video, then you extract each of
Yeah.
this mouse. So this one first.
This one, arena detection, Professor,
Yeah.
first aligned the mice,
Yeah.
and then we extracted as image here in arena
You can
detection.
use,
Yeah. After that, we have to got extracted mice in video.
yeah, this
The-
is extracted mouse. So
Yeah,
you
yeah,
want to
but
have a video acquired data, and
yeah,
you say this is video extraction.
video, yeah, video.
I mean, it's the same, but anyway, this
The
is
same,
video
yeah.
data, so video
Yeah.
extraction. And then this guy can move around, but you can extract them into, align into one. So I consider this is 1, this is 2, and this is 3. Why is this order?
Because this one, arena detection, if this is raw data, after the raw data, we
Yeah,
have to in the tool, in the arena detection tool, we have to select the center alignment. After
okay.
that, we extract and
Yeah,
then see whether it is noisy or not in here step, in the second step.
you
The
mean this
arena
extension, this one?
no, the arena detection one here
Okay,
in this step. And then in this step, we can also extract from the background. Then we
okay,
can only see the mice here, this mice. And then
okay.
after that, this one is just the video to see in the whether it is noise or not in the video section, but it is the same.
So Brano, it's very confusing. I think you should make this one into 2 slides. One slide, top one, okay? This one slide. Okay. This is about the 3D like camera and behavior open field test. That's one slide. The second slide is about like data processing.
Okay.
Okay. And then move this, put the explanation, like this simple ones sometimes doesn't make sense. You didn't put actually this is like hidden Markov. So you want to have acquired data, a bullet to explain at least 1 or 2 sentences. Arena detection, what do you mean by that? What do you mean by extraction? Why are you doing computer PCA? What does this actually mean? And this figure is actually very bad. Yeah. Nobody understands it because I cannot even see what's going on. Replace and explain the detail of this. And this is actually transition.
Yeah, transition, yeah.
You have, first of all, syllables first.
Yeah.
Where's syllables?
This one after we train AR hidden Markov model. Yeah. We got the syllables and then finally just see the visualization.
Okay, then why don't you put these 2 parts? One is a syllable, the other is transition.
Okay.
Yeah, so you have to do this one first for getting syllable and transition. Okay, visualization of syllables
Yeah.
and transition, and then you explain 1, 2. Okay, so new slide. Okay, it's 2 hours. I think that's too much. I'll just go tell you briefly. Please remake all these figures. We cannot see what's going on over here. You see, this is strange. What does this mean? Sensory metabolic instructors in this another. Is this true? 2000%?
Yeah,
weight
versus
decrease?
Control mice increase. Yeah, I did.
You mean weight decrease by 2000%? What do you mean?
I mean, it is like,
21 times?
Yeah, 21 times when compared to the mice increase.
I'm not sure. I don't understand what you are talking about this. I mean, weight can decrease. No, by 100%. I mean, this is not that much. Okay, so there's a control streptozotocin baseline. So that's control mouse. Streptozotocin, or it has a decrease. So don't do this way. Weak baseline. Blood glucose. Oh my God. You have to use like this plot. So the same animal.
Actually, okay, yeah.
Why are you comparing this between these two?
I mean, I have seen this one from the literature.
You're not supposed to this way. You're comparing that the control mouse has no differences. But streptozotocin mouse, baseline, after 2 weeks, it decreased. But you cannot identify each of the animal with this. You have to go with a paired. So the animal, I see. Don't put this way. You have to start from 0 to, let's say, like 30 or 35. Okay, we scale it. And what? Let me tell you what you have supposed to. So you have control mouse, okay, and streptozotocin, or you want to have, okay, streptozotocin is fine. So you have the dots. You have each animal. Like this, okay? Baseline, 2 weeks after, okay? And streptozotocin, you want to have. You need to make a better consistent color coding. So let's say blue, green, and it is actually quite decreased, right?
Yeah, decreasing.
So control, you probably have not significant. This one is significantly reduced. Probably this p-value, okay?
Yeah.
And importantly, you should make it this 0 because there's a concern that 5 gram. You know, animal protocol, like how many body weight decrease is acceptable or not? Do you know about that? In general, 15%.
15%, yeah.
Or severe 20% decrease, you have to sacrifice the animal.
Yeah.
The animal suffers. So I'm a little concerned. Let's say this one to here, like 29 to 22. 29 to 22. 7 gram decrease. That will be an issue. Okay. So, but data is data, but you are, so you need to understand your animal protocol for the body weight decrease. Okay, if it's too much, that is a concerning sign. Because the animal is not only sick for the food, the animal body health is an issue. Not glucose. Bone fray here. Bone fray here. Rahanu?
Yep.
This is bone fray and this is bone fray.
Yeah.
Okay. This is week baseline, week 2.
Baseline, yeah, week 2.
You cannot use different scale. You better to use the same scale. Okay. Choose either 5 so that one can see, because you see this, even thickness is different.
Yeah.
That's not good. You want to, that's why I'm keep telling you about figure panel. Panel is comparing. Whenever possible, you want to, it may look different, but you have to use the same scale so that people can compare left and right. But all of these figures need to be reworked like that, connecting. For now, given you have only 2 days, for now it's fine, but remember, people may point that out. And if you don't know, then people will say you don't know anything. Why? This is a paired. It's not different group of animal. So it has clear sequence of that animal A will decrease this, animal B may increase. So this is a more proper way of displaying. See, Bruno? But for now, I guess depending on how much you can actually spend to update, but at least this scale has to be the same. I mean, this is total difference, it's fine, body weight. Okay, the body weight. You see, and this, I don't know what, Prahano, can you explain? But this is completely wrong. People will kill you, okay?
Yeah.
Doesn't
this
make
one,
sense.
this one, like, okay, I will also use another interpretation.
Yeah,
there are different.
Please. I mean, this weight decrease is not 200%, maybe 20%. This is not 2000%.
Yeah.
Please think through and don't make the same.
Okay.
Okay. Okay, this is mosaic sequence, right? Syllables.
Yeah. Yeah.
But this syllable is not only 22, but 28.
Yeah, this one, I only extract 22 that explain 99% variance.
Okay, I see.
That also put in the I also put the graph in the supplementary.
Okay, I see. So this is supposed to be people will be taking care, paying attention to here. Please make this one big, okay? Nobody can see this. Okay.
Okay.
But I think the most important was mosaic, but this is a basic one, right?
Yeah, this is the
Position,
traditional one.
distance between mouse center and arena center. So instead of this, please make a diagram that explains that position, let's say, center. Okay. Otherwise, it's not easy to understand. Average height across the entire visible mouse. So this one, are you talking about the mouse, like
the mouse.
center?
Yeah.
Center distance from the bottom to the center of mass of the mouse. Is that true?
Yeah. From the bottom to the head.
Yeah. Please display. Make a drawing for that. Length, measure roughly across the spine. So that one's also
from the tip of nose onto the base of the tail.
Mouse length measure across the spine. So tip of tail and neck. Is it true?
The tip of the nose.
Nose. Okay.
And then the base of the tail.
Not noise. Nose.
Nose. Yeah.
And not height.
Okay.
Yeah. Please keep practicing, because you don't want to. You are public academic. You cannot use the wrong words. Okay. So then it's a L. It's H. This is a P. Okay.
Yeah.
Please make everything visual. We are visual animal. We don't understand this one, okay? You can put it there, but make this one first. So that's a position P, right?
Okay.
P. I mean, this is too much. I think you should do this one to expand because nothing, no information here. Can you actually edit this? This is so strange, right? And this is height. And speed is probably it's very easy to. And do you have any unit? So this is a pixel, millimeter. Okay, that's good. And mouse length. And the mosaic. This is completely different.
Yeah.
But I think what's very cool is the mosaic. It's a very distinct, especially this part. It's very distinct from others. So this is very promising. Why did you put this the same thickness? I think this should be expanding at least twice or several more times. This is the key. Mosaic's the most important.
Oh.
Isn't it? This is the key.
Yep. Yeah.
Why are you putting the same way? Okay, make it bigger. This one's actually nobody can see, so I can see the speed. If possible, you want to rearrange because this is no. So I don't know which one to arrange, but depending on like mosaic is one, the other, the most clear ones, length are different, very clear, right? And height is everywhere. So if I were arrange it, I would put this one at the left because it's clear. And you want to rearrange based on, let's say, this one might be also different. So height is different, right? This is majority low, but this one
height is not different averagely.
But this one at least impression is this. There's a variation. This is not that variable. I don't know, but yeah, I can see this. But overall, this the picture itself. So I would put this second, and this one also third. Speed looks very different, but you're here. I cannot see, but if you expand this one, then we can be able to see because this one certainly has some information here, and there's not much. I mean, this might be even the first, maybe second. Height is third, looks to me, from the left side. Okay, like 1, 2, 3, 4. And this positions everywhere, so it's maybe 4. But this one also a little bit kind of variable. But I mean, the alignment of this individual animal, I think it's a good idea to also align them based on some consistency. Then visually, we can see what's going on too. Align, I mean, like, you understand? This is well aligned. This is also maybe well aligned. The third one, I would put maybe longer one to shorter one, like some order, or shorter to longer, so that we can actually compare these two. So how many animals?
This one is like 12 animals, but in the 3 section, 36 total.
Okay. You better to put like kind of control.
Gravity.
So control, let's say you have, let's say maybe 4 animals.
Yeah.
3 sessions.
Yeah.
Are you putting all the different weeks or just one time?
I will do in 1 week. I will do this one in 1 week.
Okay, in 1 week, I see.
Yeah.
3 sessions during the week 3.
Yeah, during the week 3.
So it's not actually a single animal, all the sections.
All the sections, yeah.
You could potentially do like average of an animal. Do you have that data too?
Yeah, that's why I calculated the next slide average.
Because you don't necessarily put all the data first, average data.
Yeah.
No. So you probably have single animal 3 session on average. Do you have that data?
Single animal, yeah.
I mean, here, let's say this is one animal, 3 sessions, but the same animal, do you have an average for the same animal? I'm just curious.
Yes, professor.
Or,
In the same animal, in
yeah,
the 3 session, I have data.
you should have the same animal in the same next line. Here, like the same animal, the same animal, the same. This way we can actually interpret more properly. Did you have that one that way? Or
The next slide, the next 2 slides.
okay. Okay, actually, this is the hard part because people would not understand what you are talking about here. Heightened usage, no different. So it's not heightened, okay, usage. More frequent usage, right?
Yeah.
More frequent usage and then reduced. I mean, whenever possible, the same words, okay? And then no difference, no statistical. But this is less interesting, right? So you want to put here in the back.
Okay.
Frequent use of what? How many is it? 1 to the 5 syllables.
5, yeah.
Syllables. This one.
9.
9 syllables. Okay? And then no difference. How about that?
8.
But people
Totally
will,
8.
I mean, this is why is it bold. Please don't do this. Okay? You have to be consistent. Why bold face? Only this one. Is it that important?
This one I would like to, actually I already explained this one. I will change it.
Okay.
I will
So
change
the issue is people would love to know about what the wall jumping, okay, it's into head rearing. So head like rearing, head turning, nasal hesitancy. How does it explain?
Yeah, this one, like the nasal hesitancy, I put all the definitions in the.
Okay, fine. So people may ask you question, okay? And also your interpretation. Can you explain why diabetic neuropathy will reduce this and increase this? You may want to explain those. Okay, so that's like the fun part of this research though, to see whether why. Can you explain this? Okay, alterations colors. Color.
The average one, this one.
This is average one. For each animal?
On this one is
too many dots.
12 animal. I already average the 3 sessions.
Okay, so how many control? How many controls are here?
4.
Okay. 4 animals.
Yeah.
But this one looks like 12 points.
Yeah, but I put in the 12 animals, but when we calculate it, it averaged them.
I know, but that means then you would put different color coding for that. You could have done, I mean, for now this is fine, but later you should do this, try, okay?
Okay.
So put assign different colors to the same animal. So that we can see how much variations are there. the same. Or also make, instead of this, you have a mean of 4, mean of 12. Can you put it that too? So another analysis. Okay, this is just look very many, but this may not be true because people consider this point as a single animal. So this is just some way to you spread out all the points, but it's not necessarily the same animal, the different animal. They can give people, hey, this is the wrong way. Some people may say that. Or at least you want to distinguish the same animal with the same color so that people can see how consistent it is or how variable it is. That information can be put. Do you see what I mean? But the organization is very poor here because length, speed, position, height. Why are you doing this? You want to put into, in this case, I mean, this is all the same. You don't need this space pair. You just need one time. So why don't you put this one into, like, I mean, this. If it's a slide, you can put 1, 2, 3, 4. It's fine here. And then you have some interpretation. Bottom. Okay?
Yeah.
And please make this one big enough. And why this one's, why this is small capital? Be consistent. Okay?
Okay.
So, hey, Rahanu, because I don't think I have enough time now. I'll just go through and tell you. Just mention.
Okay.
Too busy slide. Okay, compare with workflow. Precision and recall. Please use the same font. You don't want to use different font here, okay? Why? This one, why are you using the same one over and over here?
Yeah, one is enough, I think.
Remove confusion matrix is one time use is fine. Just put confusion matrix somewhere and remove this. You are talking position, MOSIC, length, MOSIC, velocity, position, length, velocity. I don't see height here, but anyway.
Yeah.
What are the differences? This is okay. MOSIC. What's the difference between these?
This one,
it's the same.
I want to, no, this is the same,
Okay, so this is the line you want, precision recall curves. Precision, these are all precisions.
Yeah, for each parameter.
Identifying. MOSIC is outperforming than what? Scalar matrix?
Yep.
You mean position? So the top is about position. MOSIC is better. That's what you want. Second one, length. MOSIC is better. Third, velocity. MOSIC is better. But I don't understand why there are many. Recall is the parameter here.
Yeah.
How many recalls are here?
3.
But there are many points, data points.
This one, I want to compare like more motion sequence. This one method. The another method is the scalar method, or like
these are
including
scalar. Yes.
Yeah, position, length, like that, and then compare their true sensitive. How the rate identify the pain induced.
Why are you doing this one or the same thing over here?
True. Yeah, this one is we don't need, only one is enough to just the
don't
top
do
one
this.
is true negative.
True negative, first positive, true positive. Oh, another, please make this number big. Nobody can see. I know you're putting color coding, but that's not that important. The numbers are also better to be enlarged. Y is x, axis is 0, 1. Okay, true, false, negative, true, positive, 1, 1, 0, 0. Okay, true, negative. But you may want to put some explanation because I'm a little confused about your recall. Like, what should I do? You're talking about this one as a total average between the top and bottom.
Yeah.
But does that mean that we have parameter as a recall as many recalls? So you have a definition of recall here.
Yeah.
So this is false negative.
Sensitivity.
So sensitivity. Okay. Yeah. True disease. True positive. So different sensitivity. Okay. Sensitivity in different sensitivity. That's a precision. False positive. This is like a positive predictive value.
Yeah. Precision means positive predictive value.
Yeah.
Recall is sensitivity.
Okay. Sensitivity, PPV.
This one is F1 score. The F1 score means the harmonic mean between the two.
Okay, yeah, the mean between two. Okay,
yeah.
Anyway, please make sure you call this one, like when you say, maybe you can put like the other sensitivity and positive predictive value. And so anyway, the point is there, but this is like too much. I don't know why you're like this. And this is also like the same one, but you are comparing this way.
Yeah.
Okay, and Frobenius, no. Oh, I mean, I haven't seen this one properly, so you have to be able to explain this clearly, okay?
Okay.
I mean, people will be shocked. Remember, the audience may not have heard about this, but you are talking about symmetry, okay?
Yep.
So you probably need a little bit full explanation of this, maybe some examples, and when it's important, and reference. Okay. And then symmetry transition, you say this. Actually, this is confusing and people will ask you question, okay? Control, incoming syllable, outgoing syllables. This is this control. Actually, it's unclear to me that which one we have to focus on, but Frobenius norm is different, right?
Yeah.
So here you have to have one slide for examples for people to understand what does this number mean. Is it smaller means more symmetric or large means more? You have to explain it here.
Yep.
Put an example slide to explain clearly. Okay. People would look at it, okay, its definition, so what? You didn't explain this, whether the numbers, the explanation. So
this one in the explanation, in the upper.
So should we only see the numbers or pattern of this or color of this? It's unclear.
This one, okay.
So make sure you have to be able to, in a short time, very clearly explain this. And I think that was a little strange to me that in the end, you said once more dissimilar cluster 1, 2, 3, rearrange. This one used to hierarchical. So people may not be expert on understanding this clustering. You have to be able to explain clearly. Okay, explain what distance matrix is, and in the end, cluster 1, 2, the higher cluster 1, 2. What are the numbers? I don't see the numbers. Okay,
scale. I have used the scale bar.
Where?
The scale bar here. I didn't put the numbers in the inside of the matrix. I removed and put the scale bar.
So why are you having two slides for this?
Because this one I cannot put this one. Maybe
Oh,
I will try to put
just,
in one
just
slide.
put it in one. I mean, there's
Okay.
plenty of space. This is
Okay.
bad organization. But actually, it's unclear how to interpret this out of this figure. So you have to explain it very clearly. Otherwise, you will spend a lot of time, people get frustrated. Okay? And discussion. Figure third. I mean, this looks like a kind of future mobile. You want to summarize into several sentences. Where's your summary? Okay, that's good. Discussion is like too unorganized. But if you can explain it clearly, fine. This is not a discussion though. This is supposed to be literature, I guess.
Literature.
But neural mechanisms probably a little bit far away from, like maybe future direction. Okay, and then discussion, decision. Okay, it's good to have. Do you have all the literatures here?
Yeah, I put in the reference.
Okay, I see. Summary reduced. Okay, you want to put like some emphasis or key part, like with highlight, because otherwise, very busy slide, hard to read. Nobody can read it within like a minute. And so when you explain only that part, I would better to have something like pictures of your summary here, further study, future study. Okay, good. Histopathology. People will ask you histopathology. I mean, that's a little bit. You would rather to find out like nerve conduction velocity measurement and histopathology.
No, this one here, histopathology.
Connection. I think this further study, don't put it in the main. This future further study, put it after finishing. Supplementary.
Okay.
You don't want to put too many slides. Then you already have many concepts. It will take more than 20 minutes with Q&A. Then it may be not good. Spending a long time is not good for you. Okay. But those complicated concepts, you want to explain super clear rather than spending time elsewhere. Okay. All right. I think that's all. Now let me stop sharing.